everyone. My name is Junyan. I'm delighted to be here today in this webinar. A little bit about myself. I'm a clinical chemist at Vancouver General Hospital in Vancouver, Canada, and a clinical assistant professor in the Department of Pathology and Laboratory Medicine at the University of British Columbia. The title of my presentation today is Overcome a Big Challenge, Progress on Mass Spectrometric Analysis of Large Molecules in Clinical Laboratories. The information I will be sharing today is based on my professional experience and research in this field, and so I have no disclosures to make today. In today's presentation, I will briefly talk about immunoassays for protein quantitation. Then we will dive into protein quantitation by mass spectrometry. I will walk you through the entire workflow of protein analysis by mass spectrometry, identify the bottlenecks in clinical applications, and explore the latest advancements in each stage of the process. Lastly, I will illustrate the power of mass spectrometry through one clinical assay that I have personally contributed to. Let's start with two terminologies, protein and proteoform. Different from small molecules, protein has large molecular weight. The primary structure is the composition of amino acids. It also possesses secondary structure, which include alpha helix and beta sheet. Tertiary structure refers to interactions between amino acid side chains by forming different bonds in the polypeptide chain. And quaternary structure is the arrangement of multiple protein subunits. The complexity of protein structures result in varying levels of accessibility to different amino acid sequences. Those buried inside the protein structure are less accessible than those are exposed on the surface of the protein. Adding to the complexity is proteoform. Think of a protein as a single tree within a forest. Proteoforms are the forest itself, a collective of proteins with similar structures, but often minor variations. So this graph on the right of your screen can help you better understand the proteoforms. Within our genetic code, genes usually have multiple splicing variants. Each of these variants encode a unique protein isoform. Each protein isoform can undergo post-translational modifications, PTM, adding slight alterations to its structure. These modified isoforms collectively form proteoforms, sharing a common backbone and structure while exhibiting subtle differences. Among various tools for protein analysis, today we will focus on analytical tools for protein quantitation specifically. And we know that sandwich immunoassays are widely used for this purpose. So how does it work? The major component is a pair of antibodies, the capture and detection antibodies. The capture antibody is typically anchored on a solid phase and targets specific epitopes on the protein of interest. The detection antibody, on the other hand, attached to a different epitope of the same protein and is usually labeled with a substance that can generate a detectable signal in the final step. The magnitude of this signal directly correlates with the amount of analyte being measured. So what makes immunoassays particularly appealing is their automation. allowing for fast and high throughput analysis. However, let's recall the concept of proteoform, those unique protein variants in our biological forest. Immunoassays require antibodies to be smart enough to discern subtle differences between those closely related proteoforms. Proteoforms bring to the issue that savage immunoassays must confront, which is the cross-reactivity, susceptibility to interferences, and a lack of standardization or harmonization between different manufacturers. So what are our immunoassays truly detecting? Is the disclaimer on interferences adequate? And is the interpretation accurate? Mass spectrometry was a cornerstone of analytical chemistry in the 20th century. Thanks to the technology advancement, mass spectrometry-based approach for protein quantitation has been advancing. There are two approaches, bottom-up and top-down approach. In the bottom-up approach, we start with the intact protein, but instead of analyzing it directly, we first break it down into smaller, more manageable pieces, peptides. These peptides are then subjected to analysis through mass spectrometry. Conversely, in the top-down approach, we keep the protein intact and analyze it as a whole. Each of those approaches has its own strengths and nuances, and the choice among different approaches depends on the specific purpose of your analysis. In this table, it shows the different purposes for analysis, including identification, quantification, and modification using bottom-up or top-down mass spectrometry approach. For absolute protein quantification, bottom-up approach is the preferred method, as highlighted on this slide. Next, let's walk through the entire workflow of the bottom-up approach. The process consists of three major steps, starting with sample processing, followed by analyte separation, and finally, samples are introduced to mass spectrometer for detection. To achieve absolute quantitation, internal standards, external standards, also called calibrators, and quality controls need to be integrated into the workflow. These components provide the necessary benchmarks and references, allowing us to attain accuracy in protein quantitation. As we move forward, we will dive into each step of each of these steps, from sample processing to analyte separation to mass spectrometer detection. We will focus, and also the last part, internal standard calibrators and quality controls. We will focus on the bottlenecks that hinder protein analysis by mass spectrometry in clinical laboratories and advancement in overcoming these barriers. From a biological sample to obtaining results is not a one-stop shop. Our first hurdle in the workflow is sample digestion. The traditional protein digestion method was borrowed from proteomics studies, which involves complicated sample pretreatment, including reduction, alkylation, precipitation, and time-consuming, usually several hours to overnight digestion phase, as shown in this picture. As we strive to make these processes more practical for use in clinical laboratories, we asked two fundamental questions. Does the digestion step need to be so complicated? Can we reduce the digestion time? The paper we published in Clinical Chemistry gave you the answer. This study shows it's possible to streamline the process by reducing unnecessary reduction, alkylation step, and the peptide level cleanup. The short version of digestion now comprise only 10 minutes denaturation, followed by 20 minutes trypsin digestion before moving on to mass spectrometry analysis. The streamlined method was successfully used for apolipoprotein A1 quantitation and compared really well to the immunoassays used in clinical testing. In addition to the fast in-solution digestion, immobilized trypsin digestion can also reduce the digestion time. In this approach, trypsin is immobilized on the solid phase. As the sample passes through the column, trypsin effectively cleaves protein by increasing the contact between trypsin and proteins. This method has been successfully used for denosumab, a monoclonal antibody quantitation, with only 14 minutes of digestion. The second hurdle is the matrix effects. The term matrix effects referred to a difference in mass spectrometric response for analyte in standard solution versus the response for the same analyte in a biological matrix such as plasma, serum, or urine. These differences can manifest as ion enhancement, which is the increase of response, or ion suppression, which is the decrease of response. Ion suppression is more commonly observed, which can greatly reduce the lower limit of quantitation, LLOQ, also called the sensitivity of this method. To tackle matrix effects, we can filter out impurities in the sample in the biomatrices using different strategies. The first approach is the sample cleanup using plates made of different materials, including solid phase extraction, liquid extraction, and supported liquid phase extraction. The first sample cleanup approach applies to analyte at high to medium-high abundance. For analyte at low abundance, the affinity capture approach can be applied. It pulls down the analyte via more specific banding. These include, but not limited to, the following approaches: protein A or G banding, anti-protein or anti-peptide antibody banding, metal affinity for molecules containing histidine moieties on their surface, aptamer or polymer banding, and more recently, the label-free optical sensor has also gained a lot of interest. The selection among these different options depends on the unique characteristics and properties of the analyte of interest. The third hurdle I want to mention is the long analyte separation time due to liquid chromatography prior to mass spectrometry detection. It greatly affects the throughput of the method. When sample is introduced to the LC column, it's separated by changing the percentage of organic solvent over time. This is called the LC gradient. In addition to LC gradient time, the column needs to be well equilibrated, which typically requires 10 to 20 times of the column volume. What makes it longer is additional injection between samples. So truly, the total analysis time for each sample is the time from one injection to the next injection. So consider this clinical scenario. In a routine clinical mass spectrometry assay, the analysis time is 5 minutes per sample, and you have a batch of 100 samples. The total run time, excluding sample preparation, would stretch to over 8 hours. Now, let's compare to automated core chemistry analyzers, which are capable of processing hundreds to even 1,000 tests per hour. The discrepancy in throughput becomes strikingly apparent with higher sample volume. While protein mass spectrometry assays are often esoteric and relatively flexible regarding turnaround time, shortened analyte separation remains an ongoing pursuit. Capillary electrophoresis has emerged as a game changer in this context. Capillary electrophoresis is a routine technique in protein electrophoresis, offering significantly faster turnaround time compared to traditional gel electrophoresis. The synergy between capillary electrophoresis and mass spectrometry can be harnessed for more efficient protein analysis. The combination of those two techniques has been used successfully for therapeutic monoclonal antibodies. Now, here is the bold question. Can we bypass the liquid chromatography step for protein quantitation to be more efficient in clinical application? Liquid chromatography is simply a tool to address the limitations in selectivity of mass analyzers. So the key question becomes, can the mass analyzers alone provide required selectivity? Let's move to the next session with this question in mind. Triple quadrupole mass spectrometer is the most widely used type of mass analyzers in clinical laboratories. By its name, it consists of 3 compartments, Q1, Q2, and Q3. The sample carried in inert gas, typically nitrogen, passes through these 3 compartments. Q1 serves as the mass charge filter, isolating the peptide of interest. In Q2, the selected peptide is fragmented to smaller segments. Finally, Q3 is another mass charge filter that picks out the fragmented product. Each pair of the mass charge ratio of a peptide and its corresponding fragment is referred to as a transition. You can monitor multiple pairs of mass overcharge, which is referred to as multiple reaction monitoring, also called selective reaction monitoring. The limitations of triple quadrupole mass spectrometer primarily revolve around its restricted mass range and resolution. The mass to charge range of current mass spectrometers is up to 1500. So even when considering multiple charges on peptides, it only allows for analysis of peptides with a molecular weight of up to 15 kilodalton. Another limitation is the resolution, which can hinder the differentiation of proteoforms with minor differences in post-translational modifications. High-resolution mass spectrometry provides the highest possible precision, It includes a group of analyzers that can achieve higher resolution, and these usually include time of flight analyzers, orbitrap analyzers, Fourier transform ion cyclotron resonance analyzers, and ion mobility analyzers. Those analyzers has been used for different clinical scenario. For example, the orbitrap has been applied to amyloid typing in amyloidosis disease diagnosis. MALDI-TOF has been used for M protein identification. The mass-fix assay was developed by Mayo Clinical Laboratories that is used for multiple myeloma diagnosis and disease monitoring. The last part we're going to look at in the workflow is the inclusion of internal standard calibrators and quality controls. In quantitative protein mass spectrometry, the inclusion of an internal standard is crucial to control for variations in the various steps of sample processing. The internal standard is spec'd in to the sample as well as the calibrators. The ratio of the analyte to the internal standard is a key component for accurate calculations. The general consensus in this field include the stable isotope labeled internal standard is preferred. Those are usually carbon-13 or nitrogen-15 labeled internal standard. And the internal standard should have the same retention time as the analyte to control for ion suppression or ion enhancement, as we previously mentioned. The internal standard is to be added in the very early steps of the sample processing to control for variations in every step of the workflow. The calibrators, now there's some progress in the development of standard reference materials and reference procedures for accurate assessment of calibrators, ensuring a higher level of traceability in clinical laboratory testing. Additionally, the use of peptide calibrators has been successfully applied in some assays, particularly when the digestion step is well managed. The advantages of using peptide calibrators over protein calibrators include, first, the concentration of peptide is assigned by the gold standard amino acid analysis. And second, the batch-to-batch variability is minimal for peptide calibrators. Moreover, the availability of commercial quality control materials has significantly eased the burden on clinical laboratories to prepare in-house quality control materials. With all those advancements we mentioned, mass spectrometry has significantly expanded its role in protein quantitation. In recent years, a wide array of targeted protein biomarkers has emerged for various clinical applications, covering a range of fields, including cardiovascular diseases, such as apolipoprotein quantification, in neurodegenerative diseases, such as beta amyloid 42 to 40 ratio, in tumor monitoring, such as thyroglobulin testing, in immunological disease, such as IgG4 subclass testing. In endocrine disease, a few examples including ACTH, insulin-like growth hormone, pyrothyroid hormone, and a vitamin D binding protein. And there are also applications for therapeutic drug monitoring, including infliximab testing. Next, I will share one example among all of those clinical applications that to show you the power of mass spectrometry in solving clinical problems. This example is thyroglobulin quantification using mass spectrometry. So thyroglobulin is a large glycoprotein secreted by differentiated follicular cells. Thyroglobulin, which is abbreviated as TG, plays a critical role in guiding clinical decisions for patients with thyroid cancer. In early post-operation, measurement of thyroglobulin is used to evaluate the presence of remnant tissues and to determine if radioactive iodide ablation is required. It's also measured regularly post-operation to monitor the recurrence of disease. A challenge in the measurement of TG using immunoassay is the presence of anti-TG autoantibodies, which is shown on the left picture. Autoantibodies compete with capture or detection antibodies to bind thyroglobulin, and therefore causing falsely low results, which can be misleading in the clinical treatment of patients with thyroid cancer. Now, what's the solution using mass spectrometry? Here is how mass spectrometry offers a solution as illustrated in this graph. First, we start by digesting the sample to release the peptides. So all the proteins in the sample are digested into smaller peptides. Then we use peptide affinity to selectively pull down thyroglobulin-specific peptide. After washing and elution, the specific TG peptide are separated and detected by liquid chromatography and mass spectrometry. So it's not the intact protein is measured, it's the surrogate peptide that represent thyroglobulin is eventually measured by mass spectrometry. By digesting the sample, we effectively disrupt the interaction between TG and TG autoantibodies. This disruption diminishes the interference caused by TG antibodies, allowing for more accurate and reliable measurement. Lastly, but certainly not least, there are new tools that are designed to enhance the efficiency of protein mass spectrometry analysis in clinical laboratories. While our focus today has centered on the analytical phase, it's also crucial to acknowledge the significance of the pre-analytical and the post-analytical phases in clinical laboratories. In the pre-analytical phase, automated sample processing with robotic liquid handlers can significantly reduce the labor burden and user variation in sample pipetting, streamlining the sample preparation process. In the post-analytical phase, the review of mass spectrometry result is really time-consuming and often demands a higher level of expertise. Manual filing of results can introduce transcription errors. With the availability of commercial software solutions, these steps can be greatly improved, making mass spectrometry more practical for clinical utilization. So a summary of our presentation, my presentation today. First, we showed mass spectrometry can be really powerful and can overcome some limitations of immunoassay. There are a lot of technological advancement in this field in recent years, and this advancement and improvement will expand the clinical applications of mass spectrometry on the quantification of large proteins. Improvement in pre-analytical and post-analytical steps will enhance the efficiency of mass spectrometry, which will make it more practical for clinical application. Thank you. This is my last slide, and thank you for your attention.